I don't, for what it's worth.
Is it amino acid?
Interestingly,
Right. Many of these things have.
So it was very beneficial for...
My name is Matt Caberlin, and welcome to the Optispan podcast. Hey, everybody. Welcome to the Optispan podcast. I am thrilled to be joined today by Dr. Jennifer Garrison. Jennifer was kind enough to come out to Optispan headquarters today to sit down for a conversation. So you are at the Buck Institute for Research on Aging. Is that still the current name? Did I get that
that's
right?
the name. That's the name.
And you are co-founder and director of the Global Consortium for Reproductive Longevity and Equality. So we'll talk about what that is. You've been at the Buck now for, 12 years? 10 years?
Mm-hmm.
Okay, I feel like we probably first met when you had just started at the Buck Institute. I think I came down there for, I don't know whether it was a seminar for some reason.
I think you were on our scientific advisory
I was
board.
at one point, yeah. Maybe I was at that point. I don't remember. But I think you had just started there. And at that point, your lab was primarily focused on neuropeptide signaling in C. elegans. That's what you had done your postdoctoral work with Corey Bargman at Rockefeller.
Correct.
Right? And I think you still do research in that space, but then you also have made a bit of a transition into reproductive aging. And so we'll talk a lot about, I think, the reproductive aging part, but maybe you can just start by kind of telling us what's happening in your lab right now around neuropeptide signaling and your work in C. elegans, and if you're working in other model organisms as well.
Yeah. So when I started my lab, I really came to the aging research space thinking, basically it was a mix of my background. So I trained as a chemist. My PhD is in chemistry and chemical biology. And my postdoctoral work was really in neural circuits and behavior, using C. elegans as a model system.
And how did you make that transition? Because it's a non-obvious transition, right?
Well, I viewed my postdoc as like the last opportunity for a long time for me to learn something new and become an expert in it. That turned out not to be the case, because when I started my lab in the aging research space, that was also new to me. But I wanted to do something different, like completely different. And I kind of viewed the brain as like the final frontier in human biology in terms of like where we really don't know what's happening.
Until you got into aging.
Until I got into aging.
Realized...
But for neuroscience, I have had this long-standing interest in bioactive peptides. And that was where my PhD was focused. And when I started, exploring different labs to do my postdoctoral work in, and I sat down with Corey, and she said, these peptides just keep coming up again and again in these screens. And in general, she said that the neuroscience field considered it kind of a backwater.
Mm-hmm.
Like everyone was really focused on the prefrontal cortex, consciousness, sort of these higher level circuits.
Right.
And the hypothalamus, which is, in the basal part of the brain, was really not that interesting to most neuroscientists. But I came into it without any preconceived notions and spent a lot of time thinking about how bioactive peptides can influence physiology and behavior.
And maybe just to kind of set the groundwork, can you just describe at sort of a high level what you mean by bioactive peptide, and then what are some of the famous bioactive peptides people might have heard of?
Yeah, so they are the most abundant class of signaling molecules in the central nervous system. So that's point number one. There are literally hundreds of them, and they tend to be not essential, meaning if you get rid of one of them, either through a genetic manipulation or if you just happen to be deficient in one of them, it's not going to kill you.
Right. And is that typically because of redundancy, or?
Tends to be because of redundancy, but also because I think of them as, if you think about the nervous system from just people who are not neuroscientists, if you think about the nervous system, there's the hard wiring, right, which is like the synaptic connections where you have an action potential and electrical signaling, and that's very fast. And it's really communication between two cells that are physically connected. Right. Then you have, the sort of the connectivity at the level of like gap junctions and things like that. This is connectivity that's between two groups of cells. or tissues that aren't necessarily physically connected. And so this kind of signaling can happen over longer timescales and much longer distances. And it tends to encode things like behaviors, so things that evolve on a much longer timescale, and things that that essentially encode, I guess you could call it homeostasis, right? That's a word that a lot of people have issues
thrown
with.
around
Yeah,
a bit. Yeah.
But when you think about physiologic homeostasis, things like body temperature regulation, energy and fluid homeostasis, reproductive function, circadian rhythms, these are things that keep you alive, where you have to maintain a bunch of stuff within a certain range, and if you go outside of those ranges, you will die.
And specifically, your research has been focused on bioactive peptides in the brain, but certainly there are other tissues that can produce bioactive peptides and signal throughout the body.
Well,
right?
actually, I would call them, the reason I call them bioactive peptides and not neuropeptides anymore is because neuropeptide is really it's like a misnomer. So if you think about it, they're called neuropeptides because they were discovered in the nervous system, but they can be made and sensed by non-neuronal tissue.
Right.
So just for like non-scientists out there, the reason that they're so interesting is that they can mediate. So I think about them as like encoding flexibility in the nervous system. So they give your nervous system the ability to change function on a time scale that is commensurate with, for example, what we needed to adapt behaviors to be. So it takes a long time to evolve a new brain region. To survive, I think organisms need to be able to adapt their behaviors much more flexibly and faster than that. And so by not making them essential, evolution essentially has given your nervous system this ability to innovate on the time scale of something like a behavior in a way that allows you to adapt to your environment faster.
Yeah. And I mean, I don't, this could be completely off base, but it would seem to me also because a peptide is just like a short version of a protein, right? But
it's not, though.
Well, okay, we'll come back to that. Because where I'm going with this is that these are directly encoded by the genome, right? Unlike a small molecule.
That's right.
You can have mutations that will directly have an impact on the structure and function of the bioactive peptide. Whereas if you're modifying the activity of a metabolite, usually it's going to be more complicated than that. Probably multiple genes going to be impacted. So it would seem to me there might be a more direct way for evolution, natural selection to act on these bioactive peptides.
Yeah, exactly. So maybe an example would be useful here. And what you said just now is exactly right. For small molecules, which are also can be transmitters that signal over long distances, you have to make them with biosynthetic enzymes. So there's a bunch of proteins that carefully build these tiny little molecules that are, where you need a new enzyme to put a new moiety on a small molecule.
And oftentimes many of the most active small molecules aren't even endogenously produced. They come from bacteria or they're synthesized pharmaceutically, or you see them in our environment. So there's no way for evolution on us to act on those small molecules.
Yeah, so neuropeptides or bioactive peptides are made, as you said, just like proteins. They're encoded by a gene that makes a proprotein precursor. So to begin with, they look like proteins. And just like any other protein that is either secreted outside the cell or put on the surface of the membrane of the cell, they go through the secretory pathway. And as they're going through the secretory pathway, they actually get cut out of these larger precursors. So they're packaged into these specialized vesicles that they eventually get released from. And they're packaged with these enzymes that cut them out of the larger precursor and then modify them in ways that make them protease and peptidase resistant, because when they're released outside the cell, they're subject to breakdown by lots of different things that can happen. So they tend to not look like proteins because they have all these post-translational modifications. So I think a really good example, really emblematic of how neuropeptides function, are vasopressin and oxytocin. So you've probably heard of those. Those are two bioactive peptides. We talk about them in the same breath because they are very similar. So they're 9 amino acids. They're tiny, and they only differ at 2 positions. So oxytocin, vasopressin, they're only different at 2 positions. They are cyclized, so they're circularized via disulfide bond between 2 cysteines. They're C-terminally amidated, and they have a couple of other post-translational modifications that make them very structurally rigid and also pretty resistant to nonspecific degradation outside the cell. And they have really different functions, right? So vasopressin is also known as the antidiuretic hormone. It's really important for fluid reuptake in the kidney. There are 3 vasopressin receptors, and they're highly expressed both in the brain and throughout the body. And essentially, peptidergic signaling systems have 3 components to them. There's the peptide itself, and so the cell that releases the peptide or the cells that release the peptide. Then there's the receptor that it binds to, but oftentimes there are multiple receptors that can sense a single peptide. Many times those receptors can cross-react with other peptides. So that's true in the case of oxytocin and vasopressin. There are 3 vasopressin receptors, 1 oxytocin receptor, but there's oxytocin receptors can bind to vasopressin, and vasopressin can bind to oxytocin receptors. And so when you knock out oxytocin, for example, those mice there's almost no phenotype. And that's odd when you think about what we know about the biology of oxytocin. And that's because vasopressin can compensate. Yes. Yeah. So the pharmacology of the receptors is really complicated. But then there's a third component that no one ever talks about, which is there are peptidases that can specifically break down those peptides that can either be secreted as well, or they can be ectoenzymes, which means they're transmembrane proteins that have their active sites facing outside the cell. So that's a layer of regulation that you can get by just expressing the peptidase with the cells that have the receptor. And so it's exquisitely complicated how the signaling works. And when you think about pharmacology around these things, it's pretty impressive.
Yeah, and it seems, I think there's obviously a lot more attention on peptides now than there was 10 or 20 years ago, but still relatively understudied compared to a lot of the other therapeutic modalities that people think about, or even a lot of the other biological signaling molecules that are out there.
It's because it's complicated.
Yeah,
it's really hard. Like if you think about it from an experimental point of view, how do you correlate release of a peptide, say from a part of the brain, that's signaling to, say, a receptor on your ovary, when the receptors are expressed all over the place? And how do you think about, signaling over seconds or minutes or hours when you're measuring? So, we know so much about synaptic physiology because when you have synaptic signaling, you can literally stick an electrode in the presynaptic neuron and the postsynaptic neuron, and there is no question about what's happening there. You can measure it with really amazing precision. But this kind of signaling, it's more nebulous. It's much harder to capture. So we spend a lot of time actually building tools to study peptidergic signaling in different systems. That's been, I would say that was the beginning part of my lab.
And how did you first then start getting interested in aging?
So when I started my lab, there's a part of the brain that controls all those homeostatic systems where, when I talk about a neural circuit, what I'm really talking about is that there are groups of cells in different parts of your brain. They're all connected, either synaptically or through these sort of long-range signaling molecules. Sometimes I talk about neuropeptide signaling like the brain's Wi-Fi. So it's able to project messages that coordinate either distant brain regions or coordinate communication between the brain and the rest of the body, and in both directions, right? So, you have both of these kinds of signaling, but, there are nodes in circuits, and they all talk to each other to do different things, but the hypothalamus is really the central node for most of those homeostatic circuits. So that's sort of the control center. And when I think about systemic aging, when I think about normal aging, and I think about what happens to every older relative or friend that I have, the things that I see go wrong that aren't disease related are all focused around those homeostatic systems, right?
Yeah.
Changes in reproductive function, changes in circadian rhythms and energy and fluid homeostasis, all of that.
I was at a one of these Keck Futures Initiatives conferences.
Yeah.
This was several years ago, and Dick Sprott was there. And, I guess I'd sort of thought about this before, but he made the point explicitly, which is that, one way you can really define biological aging is loss of homeostasis. I mean, that is sort of the universal unifying feature of aging. That's sort of an unsatisfactory definition to a molecular biologist or a biochemist, but I think it is right. I mean,
yeah, there are those who would argue that I think stasis is not a great word, but the idea is simply that there are these set points that your physiology, your physiology needs to be maintained around.
Or I think what he was really getting at is the ability to return to homeostasis,
right? That when
in
you get
response to a perturbation, right? Which
pushed out
people
of,
now call
yeah.
resilience.
Yeah, when you get pushed out of range, that
Right.
there are systems in place to bring it all back.
And they work when you're
They
young.
work,
And
yeah.
they don't work as well when you're older.
Yeah, and that leads to all kinds of issues.
Yeah.
Anyway, the hypothalamus for me is fascinating because it's a really neurochemically diverse part of the brain. So unlike the cortex, for example, where there's this beautiful relationship between structure and function, where literally the cortex is arranged in layers, and you know which layer you're in because the architecture of the neurons is different between them, right? And they're named for their architecture, chandelier cells, and they have these beautiful, processes that you can literally tell where you are based on what the neurons look like. The hypothalamus is the opposite of that. All of the neurons are intermingled. They don't look necessarily different. There's some overlap, and not entirely, but there's some overlap between the circuits, meaning like sometimes a neuron that is expressing, that's important for, say, fluid homeostasis will also play a role in reproductive function. Mm-hmm. And so, it's complicated, but it's also an area that makes rich use of neuropeptide signaling. And so from my perspective, I thought, there has been beautiful work by Dongsheng Kai and others, lots of other labs, showing that there's an increase specifically in the hypothalamus and inflammation with age.
Right.
And that clearly disrupts some of those circuits. And so my hypothesis going into my lab was increased inflammation with age in the hypothalamus leads to disruption of these homeostatic circuits that in particular changes this long-range communication between the brain and distant tissues that leads to what we consider systemic aging. And so when I started thinking about aging, I was much more interested in sort of thinking through what are the more upstream points that we could consider for interventions as opposed to mitigating downstream damage that's already happened. Not that that's not important, but that was sort of just not where I wanted to focus. Right. Yeah, so that's where we started.
And you were working in C. elegans at that point?
Yeah, so mostly in C. elegans, a little bit of work in mouse, but there are some really amazing features of worms that make them, I would say, in some ways, the best model to study peptidergic signaling. I would never suggest that they're perfect models for all of neuroscience, but neuropeptidergic signaling in particular is a system that has evolved. It evolved really early. And so while there's not a one-to-one register between, there's this peptide in mammals and this peptide in worms, a lot of times that's the case. But there's similar numbers of peptides, similar numbers of receptors, all of the biogenesis, the signaling, all of the structure of the signaling systems is conserved. And so it's a great system to study something that is very tricky to get a handle on in mammalian systems. So you can learn things in worms and then apply them to other systems.
And of course comes with all the advantages of, it's easy to get lots and lots of animals. They age very quickly. So if you're interested in aging, you can actually do mechanistic experiments, all of that stuff. So
Yeah. So we started with worms, and there's still at least 1.5 people in my lab who are focused on projects in worms because we literally couldn't ask these questions in other organisms. Right. But I feel like it's important to let the question dictate the science. Yeah. And so when we can answer a question using a mammalian system, that's what we do.
Got it.
Yeah.
And then when did you first start getting interested in reproductive aging, and how did you make that transition?
One of the the project that I ended up publishing from my postdoc was that I discovered worms have a homologue of oxytocin and vasopressin.
Okay.
And I spent a lot of time trying to figure out what it was doing in worms. And my original hypotheses were that it was probably doing something like really basic, something like fluid homeostasis, something like, very ancient and conserved. And what we ended up finding was that this worm homologue of oxytocin and vasopressin mediates male mating behavior. So
also ancient and conserved.
Also ancient and
reasons.
conserved, but it wasn't necessarily where I thought things were going to go. And when I started my lab, to be honest, I didn't want to work on oxytocin and vasopressin anymore because I feel like there's just been so much work done around those particular signaling systems. And so when we started setting up lifespan analyses in the lab, we threw in the peptide in worms is called nematocin. We threw in the nematocin mutant to our lifespan experiments as a control.
And are those null for this peptide?
Null peptide mutants. We threw it in as a control, thinking like, oh, these guys are definitely going to be short-lived, right? Of course. And then they were long-lived. And that was the start.
And this was in hermaphrodites.
In hermaphrodites. Yeah. That was the start of a very long and a project that's taken a lot of twists and turns that essentially we sort of backed into this. It turns out to be working through the reproductive system. Huh. And very exciting and interesting work. So I was not interested in reproductive biology in any formal way. And it was around that time that we had a donor come to the Buck Institute who was really excited about thinking about aging through the lens of reproductive function, especially in females. And she asked whether we would start a center at Buck to study reproductive aging.
And this was how long ago? 4 or 5 years ago?
This was actually in 2018.
Okay, wow.
So 6 years ago. And I was on the steering committee for that center. Opened my mouth one too many times and became, as one does when you're too young to realize that you shouldn't do that.
Been there, done that.
And then I became the co-chair of that steering committee and really helped put that center together. And, the more you read about and understand how little we know.
Yeah.
I mean, to be honest, like as a woman, as a female, I feel like I went through what every woman goes through when you start to dig into the space, which is first I was really embarrassed, like at how little I knew about how my own body worked. Then I was horrified at how little there was for me to learn. I think of myself as a fairly curious, intellectual scientist, and I was first like, wow, I don't actually know how my ovaries work, which is embarrassing at age 40, or, that I was just starting to think that through. And then once I tried to learn, once I started to read, part of it was just trying to build the center we wanted to bring. We envisioned this center as a collaboration hub, both inside and outside of Buck, where we could bring reproductive biology and aging research together and use it as a way to think about aging in the rest of the body.
And that was really probably the first that had strong expertise in both of those areas.
It was the first center in the world to focus on it in this way.
I remember when I first heard about this, and it was one of those moments where I was like, this is so obvious, and it's such a good idea. Right. I mean, it's like, and I don't mean that in a negative way. I mean, this is something that it's sort of shocking. The field did not pick up on earlier, how important it is from half of the human population's perspective directly and the other half indirectly, but also to the extent that ovarian aging, I don't want to get in trouble by calling it a model of biological aging, but to the extent that it is, it's a really darn good one that we should be paying a lot more attention to. So
it's a human model of, it's accelerated aging in a human.
When I first heard that you guys were starting the center, I was like, oh my God, that just makes so much sense. And kind of like you, why do we know so little about this process?
Yeah,
because it's so obvious in hindsight, and yet nobody was paying attention, right?
Yeah, including us. I mean, to be perfectly frank. It wasn't something I'd ever thought about before.
And that's a pretty short time. Like 6 years is a pretty short time in biological research. So it's still early days in many ways. But it feels like there is a growing interest, more work happening in the space. Is that your perception?
I hope that we've had something to do with that.
Yeah.
When we started the center, almost right away, I would say within a year of getting that first funding to start the center, it was like incredibly obvious to me, at least, that if we were going to make any sort of impact, we had to do something a lot bigger. And, I think if we could convert the entire Buck Institute, every faculty member, every lab, every person working at the Buck to work on reproductive aging, and it would not be enough to move the needle. And so when we started the center, as I started to learn more about how big the data gap is, and it's huge. We don't understand the most basic things about why ovaries are aging faster than the rest of the tissues in a woman's body. So that's kind of the crux of the whole issue, is that if you think about, we talk about healthy longevity, we talk about health span, you look at, a graph of organ function over time, and it's not like your organs are aging exactly simultaneously.
Right.
But most of the decline is going to happen in that last few decades of life if you're lucky enough to live to old age, right? But the female reproductive system completely falls off that curve. And so from a functional perspective, ovaries are aging at least 2.5 times the rate of the rest of the tissues in my body. And that has profound consequences.
So let me dive into that a little bit deeper. Do you feel like it's a 2.5 times faster from puberty on, or is it more, like you said, it's a falling off a cliff phenomenon?
Well,
and I guess it depends on how you're defining it, right? So
Yeah.
you're defining it based on function versus sort of molecular markers of aging, but just sort of high level. Do we know enough to even know, I guess, would be the question.
I think we're still maybe wrapping our minds around things from a molecular level.
Yeah.
From a functional level, everything's clear, right? And I think for most of history, we've kind of framed ovarian function or thought about ovarian function almost exclusively through the lens of reproduction.
Yeah, so that's what I was just going to say. Maybe you can take us through a list of like the main functional measures that you would look at as a metric of ovarian function in the context of age.
So the one that's most obvious to everyone is that your ovaries are absolutely essential for making new humans.
Right.
So that's where all of the eggs are stored. So human female, we've adopted a strange reproductive strategy in the animal kingdom. Not a
Humans are weird animals.
We really
I say
are
it all the time.
Yeah, it's us, a few species of whale, and maybe a non-human primate, although that's a little controversial. Possibly some giraffes. You can count on your fingers how many species of animal go through.
So let's take a quick tangent, and maybe you can define. I think you're talking about menopause specifically.
Mm-hmm.
Maybe you can define what menopause means, and when you say only a few animals, what is it that differentiates us from, say, mice, who do become sterile as they get older?
They don't become sterile.
Or reduced fecundity.
They have reduced fecundity, which is different. So to be clear, it requires more than functioning ovaries to produce a viable offspring.
Sure.
It requires, and this is maybe emblematic of how we need to start thinking about female physiology, is that you need every part of the system to be working. You need a functional uterus. You need a lot of other things to be happening besides just having functioning ovaries and having oocytes around. But yeah, menopause is really defined as a single day in a woman's life. So it is the day on which it's defined as the day on which you have not had a menstrual cycle for 12 months. Full stop.
That's the beginning of menopause.
No, that is the definition of menopause. Everything before it is.
You mean it lasts one day?
It lasts one day.
Lots of our female viewers will be happy to hear this. I'm sure this is new information.
Everything before that is perimenopause. Everything after that is postmenopause.
That seems like a not very useful definition.
It's
but okay.
not, but so it's defined in terms of menstruation in humans.
Okay.
And it's characterized by two things. So it's the lack of a period, but it's also a dramatic change in the endocrine function of ovaries, right? So and this is something where I think people are very familiar with the downstream consequences of ovarian aging with respect to fertility and offspring producing functions. But with respect to the endocrine function of ovaries, people really don't think about that as a separate thing. But we absolutely have to start thinking about it that way. So the endocrine function of ovaries, from my perspective, I think about ovaries as like the architects of health in female bodies. So when ovaries aren't working properly, at any age, then that uncovers a whole host of different health risks.
Right.
So in like very young, say teenagers, for example, if ovaries aren't working properly, that can lead to problems with irregular periods. It can lead to issues going through puberty. Women in their 20s, if their ovaries aren't working properly, that can uncover risk for things like PCOS, polycystic ovarian syndrome, or POI. In women in their 30s, we're pretty familiar with the downstream consequences of dysfunctional ovaries with respect to infertility and miscarriages. And then, of course, women in their 40s, we're more familiar now with the symptoms of perimenopause, which are related to fluctuations in ovarian function, right? And then in postmenopausal women, when ovaries are not functioning the way they were before, that uncovers a whole host of different health risks, including osteoporosis, cardiovascular disease, all of the things that we associate with postmenopausal women.
So this is really important, and I want to come back to this, but I also want to finish, like, what is it that differentiates menopause in women from similar or changes in ovarian function in other animals, humans and a few other animals? Like, what is that key differentiator?
Yeah, and this gets kind of confusing, and I am not an evolutionary geneticist, so this is not my area of expertise, but I can point you towards some really awesome people who could talk about this for a whole hour on your podcast if you want. But there are really two aspects to it. So one is, most animals do go through a reproductive decline, meaning that, with age, most animals, and particularly mammals, have...
And also men. I mean, it happens in human males.
Well, the difference...
To some extent.
That's an important thing to say, is that reproductive aging does happen in men. The difference is that it's synced up with aging in the rest of your body.
And also, I would say probably more variably penetrant, in the sense that...
Maybe.
Well, maybe not, but actually, I'm sure the data is available. I would guess that there's a much greater variation in onset and severity in men as a function of age than there is with menopause in women. Maybe I'm wrong on that. I don't know.
I don't know as much about the male side, I'll be honest with you. So I don't want to say anything that will get me in trouble here.
We can look into that in a different day.
Yeah.
All right. So again, what is it that differentiates us from other
So we have this dramatic decrease in the number of viable oocytes. So it's not just the number, or eggs, however you want to call them. I think oocytes is more precise, but your viewing audience will probably know the word egg. They're technically different things, but with age, the number, but also the quality of eggs declines in humans. And when it drops below a certain threshold, that's when you have menopause. So those two things coincide in time and they're correlated, but we actually don't know anything about, cause and effect here.
Yeah,
so that's an important thing to say. But then there's also this dramatic change in the endocrine function of ovaries. So they stop producing all of these health-promoting hormones.
But you see that in mice as well, right? You see a change in estrogen levels.
No, it's really different in mice. So yes, mice do go through, I would say, like most animals, they go through an age-related decline in their ability to produce viable offspring. That's, I would say, pleiotropic in its and what's causing it. But they definitely do go through some of the same changes in ovarian function and egg quality definitely declines. So some of those aspects of reproductive aging you can model in a mouse, but others, like the changes in the hormones, totally different. And so that's where...
But there are changes. They're just different changes.
Yeah, well, yeah. But they continue to have estrogen, although it's different. Everything's a little different in mice. Okay. They go through, they don't have menstrual cycles the same way. They have estrus, which is a little different. Right. And so there's a lot of differences there, but there are some things that are similar, and I would say there are aspects of ovarian aging that you can model in a mouse, but there are many things that are very different. In terms of other organisms, we know that there are some species of whale.
Yeah, I've heard that. Yeah.
That go through true menopause.
And again, if you don't have an easy answer, that's okay. But if you do, I would, because I'm a little bit confused. Like, what is the simple explanation, if there is one, for how you would define an animal that does go through menopause as being different from an animal that doesn't?
It really has to do with, like, we run out of eggs.
Okay.
And we also have this dramatic change in hormones. So yeah, those are the two things.
It's more like a precipitous functional decline, and essentially it goes from, high level of function. I mean, I know it's gradual, but then you have this point at which there's essentially a complete cessation in production of eggs and a corresponding substantial change in hormonal status, which includes a pretty dramatic reduction in estrogen
Yes,
levels.
that's right. I wouldn't say production of eggs, because we make all our eggs up front in one go.
Right.
There's also, I would say, another way to characterize it maybe that might make more sense is humans have this very long post-reproductive lifespan.
Yep.
So that you can talk about overall lifespan and reproductive span.
Yep.
And in most animals, those are pretty closely coincide.
Is it though?
Yes.
I hear this a lot. Is it?
Yes. At least in terms of, in terms of the capability to produce an offspring.
But those are animals, I think, often living in the wild, where they don't actually reach old age. Like if you take a mouse in the wild, it doesn't make it past a year.
That's right.
If you bring a mouse in the lab, it'll live to 3 years. And
That's
it's reproductively active within, 6-8 weeks?
Yep.
So there's a pretty, I guess the question is, how long is post-reproductive lifespan if there isn't this fall off the cliff point?
Yeah, there's not very many animals that have that long post-reproductive lifespan.
C. elegans does, which is interesting.
C. elegans does. I mean, there's some other, I would say that's a tricky model to talk about with respect to reproductive span, but actually, yeah, we won't go into the
All right,
stuff.
We got off on a tangent. So
no, this is an important question though, and I think there are examples of whales that we know that have very long lifespans, 200 years, but that they go through menopause, a true menopause. But that on the same branch of the evolutionary tree, there's other species of whale that also live to old age, but can reproduce right up until the end.
And does reproductive rate typically fall off in those animals?
No. Well, okay. So demography of whales
that's tricky.
Is a little tricky.
Yeah.
But this is one of the grants that we funded. And one of the members of our center, Dina Amira, she is an evolutionary geneticist, and she is going to ask this question.
Yeah.
Like if you take ovaries from these two closely related species that have this very different post-reproductive lifespan, what are the differences?
Yeah, that's fascinating. And I mean, you hear the naked mole rat talked about a lot as if it is a unique outlier in the sense that the queen can continue to reproduce all the way up to to very, very old age.
That's right.
Maybe it's not as much of an outlier as people think. It's just that for a lot of these animals, we haven't necessarily looked carefully.
I agree. We have done any work. None. Naked mole rats are different, though, in that they have stem cells in their germline, and so they can continue to produce eggs through their life, right? So if you think about, if you just sort of take a whiteboard, if we just, started drawing ways to extend ovarian function, at least the endocrine function of ovaries, I'm not talking about necessarily increasing fertility to the end of your life, but if you could dream up ways to extend the endocrine function of ovaries, you can think of lots of different ways to do it, right? So like maybe you just make more eggs up front. That would be one way to do it. Mm-hmm. Or you slow down the rate of loss, or maybe you just keep making new eggs, which is what the naked mole rat does. Lots of ways to think about how to change that the slope or the end point of that curve.
Yeah.
But yeah, it's an open question. Like the evolutionary question is fascinating, and it's not just. I think you were sort of skirting around the idea that maybe it's just that humans, in the Industrial Revolution or something, suddenly jumped the gun and made our overall lifespan longer, and our reproductive span hasn't caught up or something like that. But that's not the answer.
Yeah.
I know that from talking to Dina.
Yeah, well, I think probably what we've uncovered, certainly there are more people living multiple decades post-menopause now than there have been in the past.
Not necessarily.
Well, certainly more people.
More people, maybe, yeah.
A
But we
higher
just have more
percentage
people
of
on
the
the planet.
population. Higher percentage of the population.
Maybe.
For sure. No, there's no question about
Infectious disease, yeah.
Yeah.
Probably.
But there were definitely people 2,000, 3,000 years ago that made it into their 80s, 90s, 100s,
Exactly.
right? So maximum lifespan hasn't changed.
Yeah.
But I think this probably does contribute to a growing appreciation, like you were alluding to, of all of the sort of additional negative consequences that are exacerbated by the changes in hormonal function that go along with menopause.
Yeah, I think about it as, I mean, honestly, when I think about healthy longevity in humans, like if we're really serious about extending health span, we are not going to make any progress in extending health span in females if we don't think about extending the endocrine function of ovaries, because they truly are the pacemaker for aging in female bodies. If you depending on how you want to measure it, certainly, some of the clocks, for example, show that the rate of aging speeds up after menopause.
How much does it speed up? You're talking about like the Dunedin pace?
I mean, it depends on 6 to 10% at a cellular level if you want to consider those clocks as
sure,
measuring
rate of aging,
yeah. Or however you want to say
it. But I was just wondering, is it like a twofold change in supposed rate of aging, or is it a 10% change? Or
I mean, you can. I don't know. I wouldn't put numbers on it like that. I think that's fraught.
Well, I said supposed rate of aging. Look, I'm not a big believer in the epigenetic clocks as measuring biological aging writ large, but
I think
whatever
we're not
they're
there
measuring,
yet. Yeah, I think they're measuring something, and I think that it's a worthy pursuit to try to find molecular features that will tell you something about rate of aging, but I don't know if we're there yet.
So another way to look at it would be, what is the change in risk for multimorbidity or multiple different age-related diseases?
Exactly.
That's a better way to look at it in my view.
Yes, I agree. And one way to think about it is for people who live to old age, males will spend, actually turn it around, females will spend about 25% more of their life in a period of poor health.
Defined
so
how?
Multimorbidity, age-related diseases.
Having more than one age-related disease.
And so on average, about 25% longer. So I can't remember the exact numbers, don't quote me, but I feel like it was something like 36 years for females versus 25 years.
Yeah, that sounds about right. I'm actually, that's a good number. I'm actually surprised at how often people underestimate what I call sick span, right? Which you're calling multimorbidity.
Yeah, whatever
right?
you want to call it.
But I think a lot of the numbers that get thrown out there are like, there's an 8-year gap in lifespan and health span. And I'm like, I have no idea how you're defining health span, but that doesn't make any sense. Like there are a lot more people living with one or more age-related disease than the last 8 years of their life.
Well, there are a lot more females.
So it goes up by 25% in females. And I know we'll talk about hormone replacement therapy for sure, but do we know to what extent can that be reversed or prevented by hormone replacement therapy?
Yes, we do. For women who start hormone therapy within 10 years of menopause, there is a 30% reduction in all-cause mortality for hormone therapy that includes estrogens. So from my perspective, we shouldn't be talking about whether or not someone should be taking hormone therapy. We should be talking about the risk of not taking hormone therapy.
Okay, I'm glad you said that, because that was going to be one of my questions, is like, where do you think that what should be the starting point for the medical community in terms of hormone replacement therapy? And the options are, right, start at no unless yes, start at yes unless no, or it's a completely individual sort of thing. And you know a lot more about this than I do, but my impression is that with the Women's Health Initiative, we moved from starting at yes to starting at no. And now it's kind of coming back, but it's more like some people are like, it's an individual thing. And I think what you're saying is, in your opinion at least, we should be starting at yes.
Yeah.
I mean, I get it. You're not an MD. I'm not an MD.
Okay.
Look, whatever.
Yeah.
This is not medical advice. Okay. All of the disclaimers out of the way.
Good. Okay. Thank you. It's important to say that because people want to know what I do or what I think, and I'm happy to share. What I would like to share is what the data says.
I agree completely. And I think that's more important than the letters that come after your name. And by the way, PhD means something. So, okay. That out of the way.
Yeah. So, and also to be clear, the Women's Health Initiative is a large-scale longitudinal data collection that has been going on for many decades. That is incredibly valuable. It's an amazing data set. Literally thousands of papers have been published. Studies have been published using this data set. And one of the earliest studies that was published when the results were first announced from the first cohort was a really the study itself was flawed. The way that it was, the message that was amplified by the popular press was dead wrong. And at the end of the day, there were no statistically significant results in that paper. And so everything about it was wrong. Mm-hmm. But the message that got broadcast was that hormone therapy is bad, that it causes cancer, and that it doesn't help with cardiovascular disease. And so that message, for whatever reason, really carried. And
I think there's a few reasons for that. I mean, one is human beings tend to default to the negative anyways, and we tend to be fear-based, and that created a lot of fear, right? And the medical community tends to be very risk-averse, right? So I think those two things combined. I agree with you, by the way. I mean, again, I'm certainly not an expert in this area, but the reading that I've done, looking at the literature that I'm aware of, I come to the same conclusion that you do, that we should be starting at yes with hormone replacement therapy for most women, and then the medical professionals should evaluate for that individual, is there a reason to move to no or to maybe, right? But it seems like there's now growing consensus that particular aspect of the Women's Health Initiative was overinterpreted, misinterpreted, and that led to probably, certainly in many people's view, an inappropriate conservatism in the approach to hormone replacement therapy. I saw a statistic, something like before WHI, 90% of women were on hormone replacement therapy, and now it's like a quarter or 30% or something like that. And some large number of women say they will just never do it.
Yeah, the fear-mongering, the misinformation, it's dramatic. And I think a whole generation of women have suffered unnecessarily as a result. I mean, you can read about this. We don't need to relitigate it. But there's beautiful, beautiful reviews that have delved into all of the data and all of the things around it. And I think one of the issues with hormone therapy, and I don't call it hormone replacement therapy because that, to me, hormone replacement therapy is something that generally speaking applies to women who have had their ovaries removed, or women who go through POI when they're very young, where you really are like replacing hormone therapy. I guess I don't want to call it hormone replacement therapy because it sort of implies that there's something wrong with women who are at or postmenopause, and I don't embrace that view. So anyway, semantics, but
I'll try to use hormone therapy. But
thank you.
I mean, I think words absolutely do matter.
Words do matter.
And if it gives the impression that it is fixing a defect, then that's not the right language to use.
I think it's just it enforces a negative connotation that I think is not necessary. Right. Anyway, one other thing to say about hormone therapy is when women have POI, so for whatever reason, when a young woman in her 20s has either ovaries removed or goes through cancer treatment and her ovaries stop working, or for unknown reasons, POI, their ovaries aren't working properly. When that happens and women are basically undergo menopause almost overnight when they're in their 20s, the medical community treats that like a medical emergency and brings to bear every possible weapon in their arsenal to try to combat what's happening in those women's bodies. But when exactly the same thing happens 20 years later, it's exactly the same process. We don't do anything. And we tell women to suck it up. And it's the cognitive dissonance that exists there. I can't I can't even.
You're absolutely right. Although the only thing I would add to that is we do that with pretty much every functional decline of aging, right?
I
I mean,
know.
and I mean, some of it is obvious why it's been considered normal. It happens. Some level of functional decline happens to everybody. So I think that is the explanation, but that doesn't mean we shouldn't take a much more proactive approach to delay as much as possible these functional declines that
Yeah.
are detrimental. And in the case of ovarian decline, I mean, I think, again, we haven't really talked in detail about all of the downstream consequences, but I think this might be a good time to segue to that because we kind of talked about the issue with the perception of hormone therapy. Actually, one more thing that's maybe worth saying about the Women's Health Initiative is certainly not wanting to bash it. I think you did a good job of talking about all of the value that has come from the larger study. I do think it's useful for people to appreciate, though, that clinical trials can end up with the wrong answer, right? And that can sometimes be the data itself or the way the trial was designed, or it can be the way the data was interpreted. I think in this case, it was probably a combination of both.
Yeah.
I mean, there were some issues with the design of the study, the people who were included, when they started the therapy, exactly what the therapy was.
Everything about it was flawed. Yeah.
And it was massively misinterpreted in the to some extent the scientific literature, but also amplified by the media and the general public. So it's just useful for people to consider. Clinical trials are the gold standard for sure, but they're not perfect.
No.
The other thing I want to say, though, is also this is a place where people who want to bash science use things like this to bash science. And I think you just have to recognize, not you, but you, the scientific process is really about testing models and trying to get closer to the truth, right? And very rarely do we end at a place of this is the absolute truth. And sometimes science will take us down, paths that are incorrect or inaccurate, and that's just part of the process. This does not, I don't think, a reflection on how bad science is. It's a case where the biology is super complicated. There were some unfortunate aspects of the way the study was started and carried out, and then it just got completely amplified and overinterpreted in the popular press.
Yeah. I mean, this is why we have peer review, right? There's a reason for peer review to happen. And in this case, the peer review failed. Yeah.
I mean, that happens too, right? So that's a whole different podcast we'll get into.
So many different things going on here.
So let's come back. So again, I think, I'm sure there are people out there who will push back against the idea that, we should be starting at menopausal women should start at hormone therapy being the standard of care and move off of that in individual cases.
I think that there's even evidence that starting hormone therapy before the one day when you have
that
menopause,
one day,
that in fact, the period before menopause that we call perimenopause, which can last from 4 to 10 years and can be characterized by 135 different symptoms and counting. That period of time, and that, again, this is when hormone levels, it's not like everything just goes down. That graph is an average.
Right.
Perimenopause is characterized by fluctuations in hormone levels. And there are, to be clear, your ovaries are making dozens of these hormones that are signaling to every tissue in your body. And they might be fluctuating in different directions during perimenopause, which is why we have so many different potential symptoms that can happen. But there's pretty good evidence, and these are ongoing studies, but I think evidence that starting earlier, starting when you start to see symptoms, might actually be better.
Interesting.
Because one of the, I said within 10 years of menopause, one of the things that happens if you don't have those signaling molecules around, if you don't have estrogen or progesterone or any of the other things that ovaries make, the receptors get downregulated in the target tissues. And it seems like they don't necessarily come back. So if you add estrogen or progesterone later, when the receptors aren't there, it doesn't necessarily rescue function. But again, we haven't done nearly enough to study this. Like, so many things need to still be worked out. And I would also say with hormone therapy, one of the unfortunate side effects of the WHI study, one of the major fallout, aside from women being taken off of it or told not to take it, is that for the women who do take it, they we have not innovated on what hormone therapy is, how it's administered, or what's in it since it was invented. And so what that means is that it's administered kind of like a sledgehammer.
Yeah.
Like not many options. And women then experience side effects because female physiology is complex and what one person needs is different from what another person needs. And so, I would say there's a lot of room for innovation there as well and personalization.
Yeah. I have no doubt that's true. And I think, that's also I think that's true in general of sort of hormonal changes in both men and women, right? There's very little in the way of personalization or really even sophisticated looking at the whole system,
just
right?
optimizing things.
Yeah, right. Yeah. And I don't know enough about exactly how medical providers evaluate hormone therapy in perimenopausal or menopausal women. Like maybe you can tell us, like, what do they measure?
As a perimenopausal woman, I can tell you that it's horrifying. Well, they only measure what I tell them to measure.
Okay, well, if you don't tell them anything, you're like, I'm considering hormone therapy, doc.
Yeah, so they'll look at things like estradiol, they'll look at your progesterone, they'll look, maybe if they're sophisticated, they'll probably look at AMH, although that doesn't really tell you that much. It tells you more about sort of your immediate fertility status. If they're very sophisticated, they might look at inhibin B, which is part of the feedback loop between the brain and the ovaries that doesn't really change until you're very close to menopause. And so it might tell you if you're within a year or so.
There are also many hormones that are produced by the testes or by the brain, and they signal to each other. And this is sort of a similar situation.
Let's be clear, FSH, LH.
Yeah, sure. Right.
Inhibins as well. There's A and B. Those are also produced in the male system.
Yep, absolutely.
And certainly important in ways that are different from what happened in females. Again, like there's so much that we...
Yeah, and can give you clues as to what's going on. I mean, I think this is where I was going is, when it comes to testosterone therapy in men, this again is changing a little bit. Obviously, there are a lot of docs out there that'll just prescribe testosterone. But most of the time, all they'll measure is total testosterone, which tells you almost nothing about, function. Right. And so it's quite frustrating when you realize, oh, yeah, there's all these other things that are pretty obvious to measure. And when you actually do, you can start to interpret with a little bit of sophistication what's going on. So you can start to personalize the treatment.
Yeah.
Right?
Well, okay. So to be fair to the physicians, I think, so a lot of physicians that I know, OBGYNs in particular, number one, they're very risk averse, maybe the most risk averse of all of the different subspecialties of medicine because they deal with babies and moms. And there's a lot of inherent risk there, and their malpractice insurance is very high, and they tend to be very overly cautious. But, I personally ask my physicians to order a whole slew of different
good for you.
Different metabolites and hormones and that I want to know about.
That's actually quite commendable. And I mean, that's one of the things that, we've talked about on the podcast before is that, unfortunately, in our current healthcare system, you really do have to advocate for yourself.
You do.
And so it's great that you're doing that.
But the pushback from them, and I understand this, is that they don't know what to do with that information.
Yeah.
They either haven't had the training, or the data doesn't exist, right? So that's a real problem.
Yes and no. I mean, you're right. For some things, that's true. For other things, ChatGPT can do a pretty good job. So the docs ought to know if ChatGPT knows.
I don't know. I think this is one of the things that we struggle with, right? So thinking through how do we, as much as possible, get the physicians on board to think this through?
I should clarify that. Like, I'm not trying to bash physicians. I think it's the system that they are forced to operate in, and the fact that first of all, most of these things your insurance isn't going to cover. So you have to choose if you're going to pay out of pocket for it. And they don't have the time to interpret. So I don't think it's that they don't know how to interpret it in many cases. It would take some effort and maybe a little bit of research, but they certainly could. They're smart people. They just don't have the bandwidth to do it because of the system that they're operating in.
That's right. So one of the things that we've focused on with the consortium is putting together a medical advisory board that can help us like create materials and also maybe even think through how to get the medical schools to put more information on the front end in the med school curriculum about women's health, because like the specialists who are taking care of women, the OBGYNs, the obstetricians, gynecologists, those are not the only people who should have this knowledge, right? Every single doctor that takes care of females should have some baseline knowledge about female physiology that they're totally lacking right now.
So I think I know what your answer is going to be, but I mean, it seems to me, right, that if what we've been talking about is correct, and that, for half the population, this is something that's going to happen, and there is a therapeutic modality that can have significant impacts on all-cause mortality and multiple different age-related diseases, there's really a public health case to be made for actually, making some level of standard of care better than what we've got today available to everyone. And that doesn't seem like it would be all that hard.
I totally agree. And I think the headwind from the fallout from the WHI is dramatic, right? It has persisted over decades. And I have colleagues, I have friends who are, well educated, very interested in their own health, who still tell me that they won't take hormone therapy because they're, because it's not natural and because it's going to give them cancer. And so, the misinformation is dramatic.
No, I think you're right. And again, when you get in the bubble, I'll call it the informed bubble, I hope it is. Sometimes it's hard to realize how many, you're right, very smart, educated people still don't have all of the information. And so they are believing something that is outdated.
Yeah. So that's why we're talking, right?
Yeah, right.
That's the whole point of this.
Yeah.
Spread the information.
So one question I've got, I mean, again, it seems like you are pretty strongly of the opinion that ovarian decline, failure, menopause in women accelerates the biological aging process, and thereby leads to increased risk for a bunch of different age-related diseases.
Yeah, I think I would say that like the acceleration part, I would say is still a big question mark. There's just so much work that we have to do. I think that the fact that ovaries, and I don't want to say they stop working, because I think that's also a huge misconception.
Yeah.
We know that ovaries are doing something after menopause. We know that because if you take postmenopausal women and you remove their ovaries, they have poorer health outcomes than women who retain their ovaries.
Okay.
So ovaries are still doing something. We just don't know what.
And they're still producing some level of hormones?
They are producing some level of hormones, not the same.
Sure.
It's very different. So to be clear, we don't even know what the whole cocktail of hormones that ovaries produce are. I mean, that's like a topic of active research in my lab.
And then there could be all sorts of other stuff, peptides.
Well, that's what I'm talking about.
RNAs.
Yes.
And, all this stuff.
Well, hormone just means a signaling molecule that signals over long distances, right? So that includes peptides and I think that when I think about the conversation between brain and ovaries, I think of it like a symphony, and it's complicated and there's lots of things going in both directions, and it's a conversation, and we still don't know what all the words are in the conversation, right? So the idea of optimizing hormone therapy, aside from just like trying to monkey around with the relative levels of the two things that we care about, and maybe adding in that third, because testosterone, it turns out, is really important.
About that. I think that's another place that's underappreciated, is the role of testosterone in women's health.
Oh my God, totally underappreciated. And I honestly, this is an area where I'm, avidly reading.
So let me ask you something. This is something I recently came across. And I'm wondering if you've come across it too. It's at least seems to be sometimes the case that in women on hormone therapy, that the hormone therapy itself, probably because you have more estrogen, is leading to an increase in sex hormone binding globulin, which then can titrate out the testosterone. So what you can actually get are women where you've supplemented back, right, but then you've decreased the testosterone. And I wonder, I've seen it once, but I've never looked for it before. And so I wonder how common that is.
So, gosh, if only we had a way to measure that.
Yeah, right.
Or, collect data. I would say that's a very important and open question. And what you just said is gets to the sort of the complexity of the system, right? So there's at least three different estrogens. There are probably 4 different kinds of receptors, and then there's this thing that you just talked about, which is a protein that binds to these steroid hormones and helps move them around, because they're really hydrophobic. So they're not just like floating around in your plasma. They're bound specifically to this protein. And so if they're bound to this protein, that can take them out of the circulation. There's so many layers of regulation on the system that, so just adding back the hormone doesn't necessarily help. And sometimes it can change things in ways that you didn't predict.
Right. Yeah.
So imagine for a moment that, as an individual, me, a woman, goes to my doctor, and before I start hormone therapy, and while I'm trying different things on hormone therapy, there's a panel of things that they test. So for example, like in my genome, I can look at polymorphisms in those in the receptors, in the synthetic enzymes that are important for generating or degrading these hormones. I can look at all those things and know something about my own baseline levels of this stuff, but then they can also measure these things.
Yeah.
And imagine just measuring a panel of things that would give me some idea about where I am, what I'm missing, and hopefully, having had a baseline, I would know what my own level should be.
Right.
And then tailoring the therapy to me.
Yep.
It's not rocket science.
No, it really isn't. I mean, again, I think it's important to appreciate, as you already said, that, we're just scratching the surface of all the things we'd probably want to measure.
Yeah.
But we can still get a lot of information given what we know now. And it's way more than what the average person is going to get, because they either don't measure anything or they measure one thing. Right. So, I agree. And this is something personally that I sort of regret, is I don't have any of that information on myself from when I was in my 30s and 40s. So I can sort of only look from this point going forward. But if you knew kind of what your baseline was, then, I guess it's an open question whether supplementing to your baseline as you get older is optimal.
Right.
But we certainly know that for many of these hormones, as they decline, there are negative consequences associated with that. And so supplementing to your baseline level is probably better than not doing anything.
Well, I mean, I think you covered this in one of your podcasts. Estrogens turn out to be pretty good for, longevity.
Yeah, I mean, this is something we also haven't talked about, but that's super interesting, right? So the estrogens in mice clearly increase lifespan. A couple of different ones have been tested now and shown to increase lifespan. And in humans, there's at least pretty strong correlative data that women taking estrogens have lower mortality risk. So, they seem to be pretty good for longevity. I don't think we really know the mechanisms yet.
No, God, we don't know anything.
But in people, we do a little bit, right? I mean, we do know that if you are low in estrogen, you're going to be at higher risk of low bone density and fractures.
Absolutely.
You're going to be at higher risk of dementia.
Yes.
You're going to be at higher risk. What else am I missing? There's at least 3 or 4
autoimmune
other
disease,
things, which is super interesting as well.
Cognitive decline.
Yeah.
I mean, there's so many. Cardiovascular disease is a big one. Stroke.
Sounds a lot like aging.
Yeah.
Maybe certain forms of cancer is the one place where, if anything, it might go the other direction. But I still don't know how strong the signal is there.
Well, I think that the data
breast cancer probably
mixed. I think even for women who've had breast cancer, I think the data is pretty clear that estrogen hormone replacement therapy is not going to change your risk profile.
Okay.
I think that is an area of active research. But, at the end of the day, the way that we administer hormone therapy is primitive.
Sure.
Right? We're actually giving the hormone itself when there is decades of pharmacological research And so many, we call them selective estrogen receptor modulators or SERMs. There are so many small molecule mimetics of estrogen, even progesterone and other hormones that can selectively tickle the receptors in ways and target different tissues in ways that allow you to be a little bit more selective about.
Do we know enough about that to know what
No,
to do?
because nobody has ever looked at SERMs as hormone therapy. So there is a huge opportunity for any entrepreneurs out there who, are thinking about ways to do something in women's health. This is a huge opportunity.
So just to follow that thread a little bit more, how would you think about developing one of these or testing in people? Like, what would be the design of the clinical trial in that case?
Yeah. So what would you be looking at? I mean, in women, for perimenopause and menopause, typically they measure things like reduction in symptoms, right? So the biggest ones, and there are now drugs both in the pipeline and recently approved that are approved for treating hot flashes, right? You can also measure bone density. That's easy to do. I think every woman should be getting a DEXA scan.
I agree completely.
Like, it's
You'll get
so
one
cheap.
tomorrow. You probably already had one.
I have had them because I think it's really important.
Yeah.
But there's a lot of different things you can measure as proxies for menopause symptoms. But in terms of like thinking about ovarian aging, I think ideally the clinical trials would happen earlier, like that you would want to do clinical trials in women in their 30s, where you would follow them with both looking at markers that would give you proxies for ovarian function. So that might mean pregnant, not pregnant, or, number of follicles developing, which is easy to ascertain with an ultrasound, non-invasively. You might look at hormone levels. You might look at bone density. You might look at all of these proxies for.
Yeah. I mean, the number of follicles developing seems like a pretty solid endpoint for function, and if it's non-invasive. I don't know how quantitative that is, but
yeah, you can count them.
Wow. And how sensitive do you think it would be as an age-dependent phenotype?
So, I mean, we know a lot about this with respect to fertility, right? For the assisted reproductive technologies, IVF.
Yeah, there's got to be a lot of data there.
Lots of data. And, one of the grants we funded was to Francesca Duncan to look at, so she had in early rounds of granting, she had done these beautiful studies to show that with age, the mechanical properties of the ovaries change. So they become more stiff. And that was due to an increase in collagen and a decrease in hyaluronan. Right? And so then her postdoc, who also had a grant from us, who then parlayed it into a K99 and now has a lab at WashU, she looked at whether antifibrotic agents would change output, like ovarian function, which it seems like they do have a beneficial effect on ovarian function. So then the question is, in the second round of grants, Francesca teamed up with someone who essentially, in other tissues, you can use shear wave elastography, which is a different kind of ultrasound, to look at mechanical properties. And so now they're trying to adapt that to ask whether we can non-invasively monitor ovarian stiffness using this shear wave elastography as a biomarker, essentially, for ovarian aging. So that would be really cool.
Yeah,
I don't know
absolutely.
if it's going to work, but I sure hope it does.
Very, very cool. I mean, it must be very gratifying to see these grants that your organization has given out,
Oh, I can't even tell
having
you.
an impact and people succeeding in their careers and continuing the research. That's fantastic.
There's something really, I mean, to be honest with you, this is this is my life's work. This is, I'm very privileged to have a research lab and to be able to direct science and to do experiments and to discover new things. And for as long as I can do that, I will. But this other thing that we built, it's so much more important than anything else I've ever done.
Yeah.
It's really...
Congratulations. That's a great place to be at, really. It's fantastic.
Hard to explain how amazing it's been.
Yeah. I feel sort of the same way about the dog aging project.
So, I understand.
It's kind of cool to have. It's very cool. So again, congratulations. That's really fantastic. So One thing maybe we haven't talked about, we've talked a lot about hormones. What are some other things that can impact reproductive aging? And particularly I'm thinking lifestyle. We can talk about supplements if you have anything that you think works, but
yeah, sure. I think what we know so far, so we don't know much.
Yeah.
And to be clear, what we're trying to do with the grants and with the center is to understand what is driving ovarian aging. We know so much about the downstream consequences. We don't understand the first thing about what sets it in motion or what causes it to happen, like what causes that precipitous drop so long before.
So let me ask you a question then. Do you think that ovarian aging is fundamentally different from the biology of aging in other tissues and organs?
In fact, I think for all of the pathways and targets that we know about for aging in the rest of the body, things like mTOR signaling or NAD boosters or whatever, that those are also operating in the ovaries. So to me, and again, with more data, I might change my view, but right now I think about ovaries as an accelerated model for human aging. Like, it's the same as what's happening in the rest of your tissues. It's just happening faster.
Right.
And so the question is, what is causing it to happen faster? And that's what we're looking for, those underlying mechanisms, those drivers. So for like practical advice and interventions, like, unfortunately, the same is true for ovarian aging as it is in the rest of your body. Like, diet and exercise.
Yeah.
Those
absolutely.
are the things, like,
right?
those are the gold standards
pillars
right
of
now.
Health. Yeah.
Yeah. I mean, for everything that we know about that impacts aging in other tissues, it's also going to impact ovarian aging.
So does obesity seem to accelerate ovarian
Yes.
aging?
So there's a really interesting but totally unexplored link between metabolic function or dysfunction and ovarian function. Sure. So what we know is some of the sort of more tantalizing correlations in young, healthy women. If they have underlying dysfunction in their ovaries, they're almost certainly going to have metabolic dysfunction. So women, for example, who have PCOS are more likely to get metabolic disease later in life.
And is that even if they have hormone therapy?
Yes.
Huh.
And that's something that they can, that enhanced risk, that larger risk is something they can pass on to their offspring, including male offspring. So there are genetic components here. Like, it's clearly a mix of genetics and other things. And, we need to figure out all of it.
This is sort of a weird evolutionary thought experiment, but so if it is the case that ovarian aging is accelerated biological aging in the rest of the body, is it the case that natural selection acted to cause that to happen at a different rate, or that natural selection to cause ovarian aging to happen faster, or is it the case that natural selection acted to cause aging in the rest of the body to happen slower? Does that make sense?
Yeah, I see what you mean.
I'm trying to think about what would be the selective advantage to menopause?
Yeah, I don't. Well, okay. So we're getting back into this evolutionary stuff that it's not my area of expertise, but in speaking with people who are experts, I think it's not clear. So one sort of easy to grasp explanation might be something like the grandmother hypothesis, right? That it was good to have like females around who knew sort of how to take care of babies and how to, could help and do all kinds of things that maybe the males were busy doing other things. That seems not to be, if that's an explanation, it's certainly not the whole explanation. I think in conversation with Dina and MIRA, she certainly has brought up the idea that it comes down to maternal fetal conflict, which is a whole area of evolutionary theory that I will not try to explain. But my understanding of it is that humans, so among placental mammals, of which we are one, that humans have by far the most invasive placenta, meaning, that when the trophoblast invades the uterine wall in search of blood vessels and nutrients and things like that, that our placentas are much more invasive than any other mammals. And that then sets up this conflict between mother and fetus, where there's sort of a tug of war going on. And I know that there are interesting things that happen with respect to parental imprinting. So epigenetic mechanisms that mom and dad play around with in the fetus. And so male imprinting genes tend to favor like big fetuses that grow faster and bigger. And mom's imprinting tends to pull that back. And so there's fighting at the imprinting level, and then there's this conflict between mom and fetus that I don't fully understand how that relates to ovarian aging, but
there might be some sort of selective advantage to kind of curtailing new pregnancies later in life, at least, in ancient times, right? And I guess you could imagine that, right? That if there is some value to having elder females, let's just say, in the social population, that preventing pregnancies past a certain point could come with some level of advantage.
Yeah, I mean, if you think about menopause as only having to do with fertility, then yeah, I think so.
Right. But then you think, what would the selective disadvantage to menopause be? It would at least be in part the increased risk for these other age-related diseases, but those don't show up for 2-3 decades down the road in some cases.
Yeah, I mean, from my perspective, I think we don't fully. They're clearly important and related, but I think that you can disentangle. You can somewhat separate the fertility function from the endocrine function, and I think from a fertility perspective, right, there's probably, I would say, an evolutionary advantage to not having older females go through pregnancy because, like I said, it requires more than just functioning ovaries to have healthy offspring.
And super
you need
stressful,
lots of,
right?
It's a pretty stressful state of physiology. Yeah.
So as resilience is declining, right, that's a pretty big perturbation.
It's a big thing to ask of your body. Yeah. Yeah. So it could be that, but I don't really know.
Yeah. I mean, it's sort of fascinating to speculate, but unfortunately, as is often the case with these evolutionary questions, all we can really do is speculate.
That's not true. We can do experiments.
Okay.
And we're going to do those.
Okay.
Yeah.
What's the experiment?
So, the experiment that we funded was for Dina to look at these two closely related species of whale, where they both live to almost 200 that we know about, and then to ask, what are the differences between their ovaries? Like, is it that one of them is making more eggs up front? Because that's one, like I said, there's lots of different strategies I can imagine. But what are they doing, and what's different about these two animals, and what's different? Yeah.
Yeah. Yeah. Be certainly interesting data to come. How do they actually get those samples from the whales?
That is an interesting question. So she's working with tribes in Alaska who are allowed to hunt these whales for food. Yeah. Very cool stuff.
So and then they harvest the ovaries.
Yeah.
Wow. Interesting.
I know. I'm like,
talk about field work.
How big is a whale
That's
ovary?
real field work.
Yeah.
Right?
Yeah. It's very cool.
So one last thing we haven't touched on. So again, I think the model is, at least right now, that there's accelerated biological aging in the ovary that leads to menopause, and then menopause at least partially accelerates biological aging from that point on in the rest of the body, right? Very simple model, but it's a reasonable sort of place to start. And there's some data in mice, I think, that's pretty interesting that supports that idea, right? These experiments where people have done sort of ovary transplants, right, and seen impacts on health in mice.
Yeah.
Do you want to take us through those? Like, what did they show in those experiments?
Yeah, so you can take old animals and transplant in young ovaries, and that has a lifespan extension and a health span extension too, depending on how you measure it. And so that's a pretty definitive experiment.
Right.
But then you can also take ovaries that have had the eggs removed. So this is kind of arguing for what I just said, that you can maybe separate the fertility function from the endocrine function. And when you do that transplant experiment, those animals live even longer.
Oh, interesting.
Yeah.
I wasn't aware of that.
Oh, yeah. So to me, and there's also this, I wouldn't call them experiments. There is this phenomenon that happens in humans. I don't know who decided decades ago to do this. to a human. But for women who undergo cancer treatment, so chemotherapy, radiation, obviously that kills all of the follicles in your ovaries. And so we go to a lot of trouble to preserve eggs before cancer treatment.
Right.
So that is called oncofertility. And that's so important if you want to preserve a woman's option to have biological children after she survives cancer. But for women, for girls who are too young, who haven't gone through puberty yet, they don't have any eggs to preserve.
Right.
And so what do you do in those cases? So at some point, somebody, I don't know who, some doctor decided to just take a piece of ovarian tissue. So they excise a piece of ovarian tissue, slice it up, freeze it, and then decades later, take that piece and put it back. And they don't even have to put it in the abdominal cavity. They can put it anywhere that's vascularized. So there's no eggs in that tissue. There are some primordial follicles, but this is nothing really developed. But put it back someplace that's vascularized. And in some cases, that is enough to not just restart the conversation between brain and ovaries so that they start menstrual cycles again.
Wow.
But babies have been born that way. So it turns, like, it gets their ovaries functioning again. And yeah.
I'm sure nobody's done this. Well, I guess I shouldn't say that. People do weird experiments all the time. But
Yeah.
I mean, what would happen if you did that with a perimenopausal or menopausal woman?
In the UK, there are clinics that try to sell this as a menopause treatment. So what this tells me as a scientist is that.
So hold on, let me before you go there, let me make sure I understand this. So they are marketing that they will harvest your ovarian tissue when you're young so that they can put it back into you when you're perimenopausal or menopausal?
Yeah, to be clear, I do not endorse.
Yeah, no, I get that. But I mean, it's an interesting thought experiment at least, right? Because I don't know what the answer would be, right?
Well, I think it's, even if it worked really well and didn't have like an extraordinary number of unnecessary risks involved, I still think that would not be a general solution.
Yeah.
Because what you're asking someone to do is have an invasive surgery when they're very young, pay to have tissue stored for decades, and then have another invasive surgery, which is never, so that's
but
never going to be.
you could do it on your arm.
But it's still invasive,
That's
right?
Not that invasive.
I mean, it's not going to be that is not a solution.
People are going to secret islands off of Honduras and getting gene therapy, so
I do not endorse. But what that tells me as a scientist is that there is something coming from that tissue, an endocrine factor, something that is being produced by that tissue that can get this conversation started. That tells me there's something there to be discovered.
Yeah, that's super fascinating for sure. Yeah, and I mean, so here's another question that I was thinking about, and this may make me seem uninformed about epigenetic reprogramming. But I mean, in theory, could you reprogram cells and from an adult individual and then create new ovarian tissue that then you could transplant into that individual?
That's a very interesting and open question. And I think that's one direction to go in.
Yeah.
So one direction to go in would be to take iPSCs, differentiate them, turn them into ovarian cells, maybe granulosa cells or something like that, which are the main hormone producing cells in the ovary. And then, put them behind some sort of some device that would keep the cells separate from your circulation.
There's all sorts of technologies being developed in this
and
space.
Then have that be a thing.
Yeah. I mean, it does seem like that might be a way for hormone therapy to get closer to the natural state without actually having to understand the complexity of the entire system, right?
That's one way to go. I think that's a really reasonable approach to take. There's lots of other, I think, to me, that seems over-engineered. I'm not saying it's wrong, and I think there are some companies that might go down that path. Yeah. And oh my God, I hope somebody does all of these things, like in parallel. I think that maybe an easier way to go, because, for me, like what those cells are and what they're making, that piece is the big question mark.
Yeah.
Because literally, we funded the very first single cell sequencing experiments in mouse and human across aging in the ovary. Last week, we got together like 60 or so scientists in the field at CZI to do a cell types jamboree. Because guess what? We've been collecting these data sets for years now across spatial and single cell omics. And nobody can compare data sets because we literally haven't agreed yet on what a cell type in the ovary is. That's where we are with the science. And so we're pretty far from that reality. So from my perspective and what my lab is doing is really to try to understand what's coming from that tissue. And I don't feel like that's a hard problem.
Yeah.
Right?
Yeah, I mean, it sounds like there's just so much to be done, and
so much.
So a couple of take homes I've got. I mean, one you sort of alluded to this earlier on, but it's probably worth just restating because you never know who's listening. I mean, it seems like there is a real opportunity here in terms of impact that somebody could make on human health, scientific discovery, like areas that are understudied. Creating a true sort of standalone institute of significant size to study this problem is a place where there is a need and potentially could be very, very high impact.
I don't think we need a standalone institute. I think that was the model, actually. So we built the center at Buck. It's meant to be an innovation hub. It is the place, I hope, that is, building this field. And one of the models would be to plant these centers at medical schools and universities all over the world. But then the pandemic happened. And it turns out we don't all need to be in the same place. And in fact, I think the impact is much greater to just right now to seed grants to scientists where they are all over the world. And so that's what we're doing. We give away grants to scientists. And then in parallel to that, because I don't want this to just be like, I don't know, just another foundation that gives away grants. I want this to be transformative. I want it to actually move the needle. And so giving away the grants, I think, is important. Right now, I'm working on a model where we have a second track. So we still need to continue to grow the field and give away new grants. But then they're 2-year grants. And one of the problems that I'm sure you're very aware of with science funding is the incentive structure for what we ask scientists to do, but then how we fund the science, totally misaligned. And so I've thought a lot about how do we make it so that the scientists we're funding can continue to do what we're asking them to do, which is paradigm shifting, really risk taking. Maybe it'll fail, but we want them to push the envelope and to do things that are going to lead to therapeutics and more than just products. We want true interventions. And that means that we need them to take risks. So 2-year grants are great, but that's not enough. And so, I'm trying to do a large fundraise to have a second pot of money that would be like a mini HHMI style funding, where current grantees or former grantees can apply to get what I would call sustainable funding so that they can keep doing the project that we think is important.
And I think one of the challenges is that the kind of science you're talking about is very, very difficult, if not impossible, to get funded by NIH.
Oh, yeah. So no funding for it.
You need some mechanism in place where this kind of work can continue to be funded. Hopefully, NIH will start to fund more research in this area. I think it's already happening, but
we've
hopefully
spent a lot of time talking to governmental funding bodies in multiple countries.
And again, I think you can make a pretty compelling, public health case. And, we're getting to the point, hopefully in the not too distant future, where half the legislators will be women. And so they're going to be invested. So I think, hopefully there's a place, an opportunity to actually get higher priority for
we're working on it for sure.
Women's reproductive health.
And I have to say, you introduced us as GCRLE, which is what we've been called until now. We're changing the name.
Okay.
So I think we have to stop calling them reproductive organs. I think by calling them reproductive organs, we have pigeonholed all of women's health through the lens of fertility and made it small, like a niche subcategory of medicine, when what we're talking about is half the population. So we're calling it productivehealth.org and the Center for Healthy Aging in Women.
There you go. So, and is this name change has happened already?
Yeah.
Productivehealth.org.
And the Center for Healthy Aging in Women.
Center for Healthy Aging in Women.
That's the kind of the umbrella organization.
Awesome.
Yeah.
Great. So last question. What, if any, advice do you have for young scientists, perhaps potentially young female scientists who want to get into this area of research?
Oh, I mean, email me. One of the things that I spent the last 4 years doing since we started the consortium is like trying to build out this network, which includes not just the scientists, but also the physicians, the funders, both on the private and the public side, the early stage founders, the biotech, pharma, everybody, and getting them into the same room and the same conversation. And so we need ambassadors. We need people to help us with our knowledge hub. We need all kinds of help. And in terms of, labs that are working in this space, we started an international conference every year, the Reproductive Aging Conference. We just had our 3rd meeting. So there's a lot of ways to find out who's working in this space. And I'm happy to point people in the right direction. But they can also just really literally look at it's called reproductiveagingconference.com. You can go and see who's spoken at the former meetings, and you can see who has grants from us. We've given away, 40. Well, we funded 49 scientists, but some of those are multi-PI teams. And so I want to say we've given away 43 grants total. But we're just going to keep giving away grants until somebody puts us out of business. And I hope that NIH will do that. But it takes a long time to get them to
It's frustratingly
move.
slow.
Yeah.
Awesome. Well, I wish you the best of luck. This has been a fun conversation.
Thank
I hope
you.
you've had a good time.
This is
I've
great.
learned a lot, and am very thankful that you decided to stop by. And hopefully we can do this again sometime in the future.
I would be happy to.
Awesome.
Thanks
Thanks so much. Thanks for watching the Optispan podcast. As always, if you have any questions or comments, please leave them below. If you're not yet a subscriber, hit the subscribe button, and we'll see you next time on the podcast.